Hey everybody, Mr. Murphy here. I'm going to be going through chapter 5, which is all about our different biological molecules, which we call macromolecules. So starting out, they present us a little picture here, which is a good example of a protein, which demonstrates a pretty significant level of complexity, despite the fact that the constituent parts are actually quite simplistic. When we start, just to set some vocabulary that's important, monomer, polymer, monomers join together to create polymers. And that's how each of the biological molecules that we look at are formed, from carbs, proteins, nucleic acids, and then also lipids. I just want to point out, your book tries to make a valid argument that we shouldn't really consider lipids to be the constituent parts of monomers into polymers in the same way as the others, as a lipid is really sort of a customization of a glycerol base with then different components coming off of it. However, while there are notable differences between lipids and the other macromolecules, it's just not something that I like to get bogged down on. And still thinking about the monomers of lipids as glycerol and fatty acid, I don't think that's really incorrect. The bonding of it is different. The length of fatty acid chains is different than the length of, a series of amino acids, for instance. But like I said, not something that I want to get too far into. So if we talk about it in terms of monomers and polymers, that's okay, to me anyway. So a little bit about the definitions of those things. These build up into what we call macromolecules, large molecules. Enzymes are a great example, and enzymes will come up a lot throughout this chapter and are expanded upon much more in chapter 8 on metabolism. Also, dehydration synthesis and hydrolysis are the two reactions. Now, in terms of those two reactions, I really do want to drive home that they are the precise opposite of each other. And what I mean by that is dehydration synthesis is dehydrating a molecule, taking an H and an OH out, that's a water molecule, and creating synthesizing one new larger molecule. For hydrolysis, you are taking that H and OH back from a water molecule, ripping apart that water molecule so that you can put the H back where it came from, put the OH back where it came from. So not only are they just kind of opposites, they are the exact undoing of one another. And I think that's an important point, just to recognize that these two things are just a directionality change from small to large for dehydration synthesis, or from large to small for hydrolysis. There's a little video which I encourage you to check out. We already watched it in class, so I'm going to skip over that, but it just demonstrates how these monomers come together to be linked as one new larger molecule. The video also includes the in the background kind of are the enzymes facilitating these reactions, which is a good thing to be mindful of. So we have huge diversity in the polymers that we can produce from these things, because even if you have a very narrow selection of different types of monomers to put together, since you have a variable number that you're ultimately connecting, that gives you opportunity to make just endless supply of diverse polymers. And that's why we have tens of thousands of different types of proteins within the body, even though we only have 20 amino acids, because you could have any number of those amino acids attached together in a variety of different foldable patterns, and so you can create almost anything. We start with carbohydrates. Now, carbs are our sugars, and the monosaccharide is the monomer of carbohydrates. We look at glucose, fructose, galactose as our standard C6H12O6 molecules. This talks a little bit about the way in which they can be classified, and the specific location of different functional groups, and then the number of carbons and things like that. I just want to highlight, we don't need to know these structures, particularly the linear versions of these, but I just want to highlight, for instance, glucose and galactose, which have the exact same elements, the exact same general arrangement. The only difference is that at this carbon right here in the middle, the OH, the hydroxide group, is sticking off to the right on glucose, and it's to the left in galactose. And that fundamentally changes the shape of that molecule, and therefore how that molecule interacts with biological organisms. And so we can create a meaningful difference in such a simple chemical way. But when we start applying these things to living organisms, we have to just keep in mind that these very, very seemingly subtle, maybe non-important differences can mean a whole lot depending upon how an organism is suited to react to the presence of these things. The textbook goes into a little bit about why we represent them as linear versus ring form. We'll traditionally see carbohydrates drawn in the ring form, since that's typically how we consider them being linked together in terms of larger polysaccharides and things. But this is a chemistry level thing, which is not a significant point for us. So we get a disaccharide and subsequently a polysaccharide as we keep adding more monosaccharides together. The bond between monosaccharides is called a glycosidic linkage. I bring that up, and I'd like to hold you accountable for that, just because we already know the peptide bond is the bond between amino acids. So this is the same thing, just for carbohydrates. So I think that's a valuable thing to be aware of and to recognize as we look at different molecules. And we've done this with MolView, where we take a glucose and a fructose, we do dehydration synthesis. Here's that glycosidic linkage through the oxygen here between the two monosaccharides, and that forms now our disaccharide sucrose. And it could form a polysaccharide if we continued to add on monomers to that. Okay. Now the polysaccharides, there's three big ones for us to worry about, or at least be aware of, and that is starch. Starch is the energy storage molecule for plants. Glycogen, which is the energy storage molecule for humans and for animals. Well, really, mammals, animals. I can't be confident in all animal phyla, the extent to which glycogen is used. Doesn't really matter, though. The point is that these two are very similar to each other. They are branching molecules. However, you'll notice with the glycogen, which is found in animals, it's far more branching. And the reason for that is because the enzymes responsible for liberating glucose from these molecules, remember, these are energy-storing molecules. In a plant, it has a few branches. And so enzymes can come to the ends of those branches and pick off glucoses as the plant and its cells need that energy. For an animal cell, where perhaps you need a lot more sudden energy, you have a lot more instances of sudden demand, as an animal's running away from danger or doing some challenging task, and you suddenly need a lot of energy. Having more branches, highly branched, just means you have more places for that glucose to get picked off. And that's why if you become suddenly startled, you feel that rush of sugar that's being dumped out. And adrenaline is leading to the release of this sugar from your glycogen in your liver. It's being dumped into your bloodstream, and you feel that almost immediately because it's so highly branched that it can act very, very quickly to liberate those glucoses, get it into your bloodstream, and get it to the cells throughout your body that might need it. The third type is cellulose. This is the major tough part of plant cells, and you'll notice that similarly its structure indicates what its function is. Here we have these tendrils of just long chains of glucose, and it really creates this kind of rope-like pattern of just strong, bonded together glucoses with a lot of glycosidic linkages that help to hold the chain together. Hydrogen bonds existing between the individual chains just really provides a lot of strength. And it's very difficult for animals to break this down because, again, the enzymes have to work from the ends of these molecules, and so you don't have nearly as many ends on cellulose as you do in the more branched structures of starch and the highly branched structures of glycogen. So it's a little bit more about the linkage of cellulose. Okay. And a little bit more information about the inability to hydrolyze that. Chitin is a fourth one. I don't go into much detail with this. I won't be using this as an example myself. However, it is basically the fungi analogy to cellulose. It's a different structural support molecule which gets used for the exoskeleton of arthropods and then also in cell walls of fungi. Lipids. So, like it says, they don't form true polymers. It's okay to consider them monomers and polymers, though. We have glycerol and the fatty acids, which are the quote-unquote monomers that are coming together to form them. A triglyceride, then, is a glycerol with 3 fatty acids coming off of it. because that's like our traditional way of introducing lipids, so that's why we follow that. But I do want to highlight that most of the lipids that we're going to be talking about are phospholipids surrounding every cell and all membrane-bound organelles. And so I want to show you what that looks like. Before that, just a quick difference about saturated and unsaturated. Saturated is saturated with carbon-hydrogen bonds, and so it has all of those carbon-hydrogen bonds throughout here. It's a very straight, simple structure. It condenses more easily. It solidifies more easily. That's how you can remember, like animal fats, the ones that, stay solid, like butter, like the fat on a piece of steak, meat, whatever. Meanwhile, plant oils are the ones that have unsaturated bonds within them. It gives them an irregular shape. It makes them less likely to settle down into a solid until they're at a much lower temperature. So that's why they stay liquid. Now, as I said, I wanted to introduce, let's see, I think we probably know this, but one primary function is energy storage, and then also warmth and insulation in adipose tissue, which is fat tissue. But another absolutely significant function of these is the phospholipid. And the phospholipid is two fatty acids and a phosphate attached to a glycerol. And so that's why there's two fatty acid tails and then that phosphate group that gives it that hydrophilic nature at the top. So that's the predominant way in which we're going to see lipids coming into play with a lot of the topics that we look at, because we're going to see membranes everywhere. All of biology is happening across membranes and differences that get established across these membranes. So we'll see a lot about that, and that's where lipids will kind of shine for us. Okay. A little bit more about the characteristics of those. The term steroid and cholesterol, just understand that those are lipids. We'll talk more about steroid hormones, for instance, and it's important then to remember that because they're lipid-based, that gives us some information about their ability to traverse the cell membrane. So just being generally aware of the different classes of lipids is helpful, but not something that's going to be extensively tested in this class. All right, and then we come to proteins. So as this says, proteins account for more than 50% of the dry mass of cells, which is kind of crazy. Proteins are everywhere. And I really try to drive home the fact that when we think of proteins outside of the scope of biology class, we probably mostly think of like muscle, right? But that is one tiny little subset of what proteins are actually doing in our body. Proteins are the workhorse of the cell. We talk a lot about how, the nucleus controls the cell and things like that. And sure, the DNA is the instructions, but the DNA can't do anything except tell the cell which proteins to make, and it's the proteins that are actually doing all the jobs in the cell. So we see enzymes, obviously, we talk a lot about those. Antibodies are defensive proteins. We have different storage proteins. We have transport proteins in the cell membrane, hormonal proteins. We have receptor proteins also in the membrane. Contractile and motor proteins, that's like your actual muscles and what you think of, although your muscles are actually tissue made out of many different muscle cells, and those muscle cells then contain these contractile bands which are doing the actual contracting and such. So lots of different functions of proteins. Don't get trapped in just thinking of them as a muscular thing. So when we talk enzymes are catalysts. They speed up chemical reactions. That's certainly important, and we'll get into that a lot more in a later chapter. But any protein is really just a series of amino acids that are chained together. I use the analogy of, if I were giving you Lego bricks, and I gave you 20 different kinds of Lego bricks representing the 20 amino acids, but you could have as many of them as you want, and then you consider, okay, well, what could I build? Probably anything, right? As long as I gave you some diversity between those 20 different blocks. The point is that if you can put these together, as many as you like, we can probably build some pretty complicated structures. And that's true of proteins. So we can get thousands of different functions out of the same basic 20 ingredients because of the order in which we place them and the number of each constituent of the 20 different types that we put together yields a uniquely folding thing, no matter what the sequence is. Every sequence is going to be unique in terms of the outcome of how it folds. So here's our 20 amino acids. We don't need to memorize those, but I do think it's important to know that we have our nonpolar amino acids, we have our polar amino acids, and then we have our acidic and basic ones, which are negatively or positively charged, respectively. And because of those characteristics, they're going to want to be inward-facing if they're nonpolar, outward-facing if they're polar. They're going to want to attract together positive and negative. Or there's the one special case here, cysteine, which is going to want to create a disulfide bond with another cysteine, which is part of our tertiary structure, which can really add stability to the three-dimensional shape of a protein. We call the bond between these peptide bonds. It's another example of dehydration synthesis. As you can see, polypeptides could be just a few peptides long or 1,000 monomers in a row, 1,000 amino acids in a row. When we look at the different structures of it, we want to be mindful of a few things. First off, that structure is created by interactions between all of the different amino acids, not just with the surrounding amino acids, but the entire structure's worth of amino acids and the environment around it. And so that creates really complex three-dimensional shapes. Here's an example of a protein from a flu virus, from an influenza. And you can see that the antibody that the body has produced has a complex three-dimensional shape, but it actually has this region, which is this like exact mirroring of the protein from the flu virus. So the shape of these things is paramount to their functionality. And for an antibody, it's going to want to be able to grab onto something that it has matched as a foreign invader. And so the function here is that it has this region that matches the shape of the flu virus, so that way it can bind and then signal, presumably, the body to eliminate it. But there's 4 levels of this, and these are all happening simultaneously on top of each other. It is not like a series of events or anything like that. It's not a step-by-step process. It's just 4 different types of interactions all at the same time producing the structure of a protein. Now, the primary structure is a little different because the primary structure is really just a matter of what did the DNA tell the ribosome to make, because that's the order of the amino acids. And so you can have this big long chain here that the mRNA told the ribosome to just put all of these different amino acids in this particular order. That's the primary structure. Then, as soon as the ribosome begins putting these together, by the time it's a few amino acids in, we start getting the secondary structure. And the secondary structure is caused by hydrogen bonds between the actual amino acid, the repeating blocks there, the carboxyl group and the amine group of the amino acids. And so this can result in typically 2 different types of shapes, an alpha helix or a beta pleated sheet, where basically, this alpha helix picture here, you can see that this amino acid, its amine group, is ever so slightly attracted to the carboxyl group on the other side. And so we have this attraction. And these are, 1, 2, 3, 4 maybe amino acids apart from each other. So like 4 amino acids down on the chain of the primary structure, we start getting interactions that cause this coiling. Similar, although different shape arrangement for the beta pleated sheet. But the point is that this secondary structure is caused by interactions, hydrogen bonding interactions between the carboxyl and amine groups of different amino acids, local to each other. Here's a spider. The tertiary structure then is caused by interactions at the R group level. So this is where those 20 different amino acids and what their characteristics are really dictate the three-dimensional large-scale folding of these proteins. And so you could see a series of different bonds resulting in these long chains folding back on each other. The quaternary structure results when you have 2 or more polypeptides interacting with each other. So this is not every protein. Some proteins are a single polypeptide chain and that's able to do its job. But some, like collagen here, has 3 different polypeptides that intertwine with each other and form kind of this fibrous rope, which is important in connective tissue. Here we have hemoglobin, another great example where you have the different subunits of hemoglobin. There's 4 of them that then become associated with each other with a little iron atom between them. And that's important for the blood carrying capacity of the oxygen carrying capacity of our blood. This goes into some other examples related to that. So these are examples of proteins and how they become arranged by the different levels. Now, similarly, they can become unarranged or change their arrangement because of influence by exterior or external factors like temperature and pH, the presence of other molecules nearby. And we call this denaturation or denaturing, which is just simply the change in shape of the protein. When a protein is denatured, we consider it to be inactive. So we may see an influence on a protein, whereas you gradually change the temperature, the protein gets less and less effective. When it reaches that threshold where the protein's not able to do its job, we now consider it to be denatured. It's not necessarily permanent, because as long as that protein does not break its polypeptide bonds, the covalent bonds, then as long as we shift those environmental factors back to normal, that protein will most likely resume its original shape and therefore function. However, these things can be fatal. This is why fevers are so dangerous. It's also why fevers are effective, because our body responds to a potential invader by raising its temperature in the hopes that whatever microorganism might be affecting you will be much more susceptible to failing to be able to accomplish its tasks at a slightly elevated temperature. But hopefully your fever's not so high that it actually prevents your own proteins from functioning properly. So, as I said, it can be generally reversed, but not always. If it's too much, it's too much. If you boil the proteins, you're gonna fundamentally break and alter them in ways that can't be bounced back. So here you can see a diagram exploring that. Denaturing into weird shape, this is pretty extreme denaturing, and then renaturation when it goes back to its original shape. Okay. There's a lot more detail about the history of the identification of these different things. We'll get into some of that in a later unit as we talk particularly about DNA. And then the last section of this goes into nucleic acids. The only thing I'd like to say about that is you should be aware that the DNA and RNA are the polymers of nucleic acids, and that they are made out of the monomer nucleotides. Beyond that, we're going to have an entire unit all about the DNA and RNA and nucleic acids. So we'll get into all of this at that point. So we will sort of wrap up this chapter with carbs, lipids, proteins, broad understanding of macromolecules, a little bit of details about nucleic acids, but otherwise we'll leave the rest out. Okay. If you have questions, please let me know.